Hello, everyone. Thank you for being here. I'm Sylvia. I'm a graduate assistant at the University of Nevada, Las Vegas. I look forward to discuss our paper, Genome-wide Clustering on Integrated Chromatin States and Micro-C Contacts, which reveals chromatin interaction signatures. Before we dive deeper, let's define a few important terms and research backgrounds that informs our study. Chromatin states or chromatin interactions are when two regions of the genome comes in contact. They're important for regulating the gene expressions. And chromatin states are regulatory annotations defined by patterns of chromatin marks. High C and micro C captures the 3D chromatin conformation. The common issue with integrating the chromatin marks from the high C data is because of its larger window size, which could be about like 5 kb windows. And it is harder for you to correlate with the chip sequence peaks and etc. So integrating this 1D chromatin state with the 3D contact data is challenging, and our study proposes a method to integrate and cluster this information across the genome. So our goal here is to provide a new layer of genome annotation beyond traditional chromatin states. So this figure here shows the distribution of chromatin states that each region is in contact with. It shows the distribution varies greatly between each of the cell types. So our methodology of creating a sum of chromatin states by contacts. So here you can see three different regions, region A, B, and C, where region B and C are both annotated with enhancer A1, and region A is annotated with transcription start sites. So the arrows between this region are nothing but the KR normalized micro-C scores. So the KR normalization is a method used to correct for biases in the micro-C data. The score should be the higher the scores, the better the contacts are. So you can see for region A across the rows, and you have the columns which corresponds to the contacts which it has. So region A has contacts with in B and C, which is enhancer A1. So that's what you see here. And you have region B and C, which contacts the TSSA. So it is in the column TSSA contact, and then the rows corresponds to region B and region C. So it summarizes this kind of generating a matrix can summarize the complex network of interactions into a format that can be easily analyzed using like clustering techniques. So our basic goal here on the ASCS matrix is to capture both the strength of the interactions, that is using the micro-C scores, and the types of chromatin states in which it has contact with. So this is a real interaction in the genome. So this is how it actually looks like. Here you can see that it is just 1 kb micro-C window, but it has so many interactions between multiple labels. So this can be really complicated to study. And this is where our ASCS matrix comes to play with. And it kind of helps us to design like more complex data into an easy form of matrix. So this is a full ASCS matrix, which summarizes the micro-C contacts weighted by chromatin states for each of the genomic region. So here you can see a specific, and then you can also see like a diagonal intensity, which means that the same region contacts with the same labels. So that is why you have like higher intensity towards diagonal regions. So based on this observation, we employed an unsupervised clustering, which characterizes the patterns of chromatin interactions. So this is the clustering of ASCS matrix. So the first part here is the contact region. So the contact region is like in essence shows the enrichment of contacts between the different chromatin states in each of these clusters. So we got about 18 clusters, and the one on the right shows the base region, which kind of tells you the annotation between the contact region and how much percentage of a specific contact is there between each of the clusters. So you can see that there is a higher intensity towards this region. For example, we can take transcription start site from the flanking region and how much of it is present between transcription start site versus the enhancers, start sites versus the enhancer gene, and the percentage of that region being present. So what we observed is about 12 to 40% of these regions change in cis between each of the cell types, but they did maintain the same chromatin states. So the cis changes at transcription start sites associated with gene expression. So the figure A shows the significantly upregulated HFF genes, and figure B shows significantly downregulated HFF genes. So in the upregulated HFF, you have more contacts with the enhancers and enhancers in genes. When we look at the HFF in cis contacts, but when we looked at the downregulated part, we could see that there is more number of bivalent and more number of repressed polycomb state regions. Uh, so the thing is, cis alone could provide about 75% of the information that chromatin states provides about the gene expression changes. So combining the cis and the chromatin states gives us more information than just the chromatin states. So although cis shows less amount of information dependent on the gene expression, kind of helped us to quantify the entire pattern of the expression change. So we also did a case study of NANOG gene. So NANOG is a well-characterized pluripotency gene in H1 embryonic stem cells. So we took contact score greater than 0.015. It is a threshold which is chosen because the contacts above this threshold are considered strong enough to be biologically relevant. And you could see that this is the region of the NANOG gene, and you could see that in H1 there was higher interactions for this specific gene, whereas in endoderm that there was no, the contacts for that region was very low, which were the overall pattern supports that the observation of NANOG has more enhancers in the H1 compared to more enhancers in the endoderms. So this was consistent with our higher gene expression, which we also performed. To summarize, CIS integrates 3D contact and 1D chromatin state data. It provides genome-wide view of chromatin interactions. It reveals patterns not apparent from chromatin states or contacts data alone. It allows identification of regions with regulatory potential. To make it easy for you to review our work later, I provided the QR code linking to the full paper. I'd like to take a moment to acknowledge the authors of this paper, Dr. Kareen, Dr. Dylan, and Dr. Mira. And also, I'd like to thank our dedicated lab members at the HON Lab and the ASCS for providing this opportunity. Thank you.